Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observation and commentary on the JCO article, Off-the-Shelf Viral-Specific T-Cells to Treat BK Virus, HHV6, CMV, EBV, and adenovirus infections after allogeneic hematopoietic stem cell transplantation by Zanu et al. My name is Sarah Nikiphoro, and I'm a clinical instructor and medical director of the Cell Manipulation Core Facility at Dana-Farber Cancer Institute and Harvard Medical School located in Boston, Massachusetts in the U.S. My oncologic specialty is stem cell transplantation and other cellular therapies. In this podcast, we will discuss the clinical need for antiviral therapies in immunocompromised patients and the rationale for using T-cells as therapy. I will review how the manuscript by Zanu et al. Reflects decades of work on expanding small numbers of viral-specific T-cells from healthy donors to create potent cellular therapies. And I'll leave you with thoughts on how this approach to adoptive T-cell therapy, which does not involve genetic engineering, may be applied more broadly against cancer cells. As a reminder for those of you not in the immunotherapy field, infections remain a well-known and feared cause of morbidity and mortality in immunosuppressed patients after allogeneic stem cell transplantation. Improvements in transplantation outcomes over the past 30 years have been partly driven by administering prophylaxis against infectious agents like varicella-zoster, and partly by early detection of viruses like CMV, cytomegalovirus, and Epstein-Barr virus, or EBV, before end organ disease develops. We now have access to drugs such as cidofovir for adenovirus and foscarnet for human herpes virus 6 infections. However, many available antiviral drugs carry significant toxicities, such as bone marrow suppression or kidney damage, and some viruses, such as BK virus, which causes debilitating hemorrhagic cystitis, have no even moderately effective therapies available. Despite the above modalities, these viruses remain problematic after standard allogeneic transplantation, with over 60% of CMV seropositive recipients reactivating CMV and around 16% of recipients experiencing BK cystitis. Newer trends within transplantation using alternative donor sources like umbilical cord blood stem cells and using T-cell depleted grafts to prevent graft versus host disease are associated with a higher incidence of infections. So the need for effective antiviral therapies in this immunocompromised population is real and unlikely to diminish anytime soon. The major precipitant for viral infections after transplantation is lack of effective T-cell surveillance. Therefore, a logical approach is to add back cytotoxic T-cells that recognize viral proteins. One way to generate such a therapy involves selective stimulation and expansion of antiviral T-cells that are already present at low numbers in healthy people. Most adults have been exposed to CMV and EBV, for example, at some point, and thus can be donors for these types of cells. Please note that genetic engineering is not necessary in this approach. Starting in the late 1990s, multiple groups headed by Philip Greenberg and Stanley Riddell at the Fred Hutchinson Cancer Center, Richard O'Reilly at the Memorial Sloan Kettering Center, and Helen Heslop and Cleo Rooney at St. Jude's provided proof of this concept by using polyclonal mixed CD4 and CD8 positive CMV or EBV specific T cells in clinical trials of between 5 and 36 transplant recipients. They demonstrated excellent safety profiles and lasting reconstitution of antiviral immunity. These T cells, when infused, decreased rates of viral reactivation when used as prophylaxis immediately after transplant and showed responses in active CMV disease and against EBV post-transplant lymphoproliferative disorders. However, these initial studies used cells generated against a single virus and took months to generate. The labor-intensive manufacturing entailed establishing CMV-infected fibroblasts or EBV-infected lymphoblastoid cell lines as sources of antigen, and then multiple rounds of complex co-culture and expansion. Each T-cell product was manufactured for a particular patient, typically from his initial stem cell donor. Notable limitations here were expense, time, and the expertise of manufacturing needed. At this point, antiviral T-cells were not a widely exportable therapy. So in the 20 years since these initial studies, efforts were devoted to streamlining manufacturing. Using dendritic cells pulsed with viral peptides or transduced with viral vectors as the source of antigen dramatically diminished production time. More recently, antiviral T-cells have been expanded using only peripheral blood cells, commercially available viral peptide mixes, and a limited cytokine mixture. This culture process can now be completed in 10 days, although testing for microbial contamination often adds another week before actual infusion. There are even 2 studies generating EBV-specific T-cells using just an overnight stimulation with EBV peptides. This is then followed by selection of activated viral reactive T-cells based on their secretion of interferon gamma. Building on these manufacturing improvements, research efforts next turn to expanding the number of viruses detected. While CMV and EBV were initially targeted, antigenic peptides from adenovirus, human herpes virus 6, and BK virus have been used to generate reactive T-cells. At least 6 clinical trials involving mixed T-cells reactive against CMV, EBV, and adenovirus, those 3 viruses, have shown efficacy as both prophylaxis and treatment without significant complications. Each T-cell product may individually recognize 1, 2, or all 3 viruses. This is dependent partly on whether the donor of the T-cells was previously exposed to each virus. In 2014, a phase 1 trial of T-cells targeting those 3 viruses plus human herpes virus 6 and BK was published. Researchers then asked whether a frozen T-cell line, originally manufactured for one recipient, could be utilized in other individuals. In 2002, Drs. Hake and Crawford established that EBV post-transplant lymphoproliferations could be treated with frozen and thawed EBV-specific T-cells that were not fully HLA matched to the recipients. Subsequent trials have shown that frozen antiviral T-cells derived during the initial trials can again be effective in recipients who share as few as 2 HLA alleles with the T-cell donor. There is a caveat here, namely that selection of a proper T-cell product is also dictated by whether the HLA alleles in common between donor and recipient can actually present peptides from the virus of interest. With the preceding discussion, readers should now be able to see how the companion article by Zano et al. Using previously banked or, quote, off-the-shelf, unquote, T-cells to treat BK virus, HHV6, CMV, EBV, and adenovirus infections after allogeneic transplantation represents the culmination of these many advances. In this phase 2 study, a bank of 59 frozen T-cell lines generated to recognize CMV, EBV, adenovirus, HHV6, and BK viruses was used to treat 38 patients with active viral infections. 7 of these patients had multiple concurrent infections, and most had failed standard antiviral therapy. Overall response rate was 92%, and rates for individual viruses ranged from 67 to 100%. The safety profile was excellent, with no significant graft versus host disease or cytokine release phenomena. Viral-specific T-cells persisted for up to 12 weeks, despite being only partially HLA matched. And notably, of the 56 patients screened for the trial, 54 had a suitable line identified. This emphasizes the relevance of these banked cells for a large segment of the eligible population. One caveat to using partially HLA-matched T-cells is that they do not engraft or provide long-term immunity, as did the fully matched recipient-specific cell lines used in initial studies. Therefore, multiple doses may be necessary. Given these results, randomized trials employing antiviral T-cell banks are in planning and commercialization is being contemplated. Finally, to whet your appetite for advances to come, the clinical applications for such antigen-directed T-cells are growing. This same group published a trial using trivirus-specific cells in patients with primary immunodeficiencies, thus expanding the eligible population. T-cells specific for EBV and human papillomavirus proteins are in clinical trials against virally driven lymphomas and head and neck tumors, providing an entree into their use in cancer. And in what may portend an even wider application of this type of cellular therapy, cytotoxic T-cells specific to tumor antigen can also be expanded using 10 days of specialized culture. These antigens include NY-ESO-1, the MAGE proteins, PR1, and WT1, among others. Again, this is without genetic engineering being required. Such tumor antigen-specific T-cells are currently in clinical trials for lymphoma, myeloma, and other malignancies. Thus, work on adoptive T-cell therapies such as that presented by Zano et al. Will continue to yield new approaches to antiviral and anti-cancer immunotherapy for years to come. This concludes this JCO podcast, and thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.